Welcome back. Now we will attempt last two questions of 2021 and this is on PCR. Which of the following is not an application of PCR? Detection of gene mutation, molecular diagnosis, gene amplification, purification of isolated protein. So PCR is related to DNA. Therefore, this will be the answer, option 4. Next one, during the process of gene amplification using PCR, if very high temperature is not maintained in the beginning, then which of the following steps of PCR will be affected first? So high temperature helps in denaturation of DNA. So denaturation step will be affected. Option 4. PCR stands for Polymerase Chain reaction. It has 3 important steps. Number 1, denaturation step. The DNA is double-stranded molecule. The 2 strands are held together by hydrogen bonds. So high temperature around 95 degrees centigrade, this is going to break the hydrogen bonds and the 2 strands will separate. Okay, so the sugar phosphate bonds are not broken, only the hydrogen bonds between the nitrogenous bases. Adenine pairs with thymine with 2 bonds and guanine with cytosine with 3 bonds. So high temperature only breaks hydrogen bonds, resulting in single-stranded DNA. So once we have single-stranded DNA, then the temperature is brought down around 72 degrees and then the primer, which are short oligonucleotides, they complement with each of the separated single strands. Temperature is brought to around 58 degrees centigrade depending on the type of primer, and this leads to extension of the primers. So at the end of extension, 2 strands of original DNA now becomes 4 strands. And then the cycle is repeated. So this is repeated around 30 times, 30 cycles of PCR, and we end up with around billion copies of DNA. These are NCERT references. So PCR is basically DNA replication, but it is happening in vitro, outside cell. Multiple copies of the genes are produced using primers, which are chemically synthesized oligonucleotides complementary to the region of DNA. The enzyme used is DNA polymerase. This is isolated from Thermus aquaticus, and this is the reason it can tolerate very high temperature.